All right, so what we have discussed so far over the last 3 weeks has been the phrases or the content that would make a really great title, right? So when you're writing a lab report, you have to implement a title that is going to catch the attention of the reader and provide some or specific information about what the study is about. So that's going to be important too. But you don't want the title to be so long and laborious that you're going to lose the attention of the reader, right? So it's this really fine balance. And so we've talked about those components, and they usually will have something to do with the objective of the entire study. Some people also like to throw in the overall approach that is used in the title. And I just want to also remind you that a title can also be the takeaway message too. So keep that in mind when you're writing your final title for your final report. Do you want to focus it around the objective only, or do you want to focus it on what you found, but you should still probably incorporate these terms? So totally up to you. And so I just also want to remind you that a writing workshop isn't going to tell every team exactly the words to write in the exact order. There is some variation, and it just kind of depends on writing style. It depends on what you and your team have discussed is in regards to results and what you think they mean. But I'm going to hopefully in this class provide you with a little more guidance than what I already think I provided on the expectations, but why not? Okay. So the other thing that we've talked about in class is some of the predictions, and we know that this has to kind of be established early in the study so that we can actually do the experiment. But this type of information in a very concise way would probably appear in the introduction of your report. And so we also talked about, so I'm actually going through and grading some of the TLC reports, and I just want to kind of PSA, the stuff that we talk about in this workshop or in writing workshops in Zoom Lab, if I say, hey, this is how it should be written in the report, I really mean this is how it should be written. If I say avoid phrases like, then really do avoid those phrases. So I'm reading not a lot, I have just started, but I'm reading some of the TLC reports, and teams are losing points because they are literally adding in the stuff I said not to add in. And then it makes me wonder, why do I even host these writing workshops if no one's even listening? Why do I record this stuff if no one's going back and watching the URLs? So please do make sure that you're taking really good notes. And when I say, is there any questions about what you should add or not add, say them now, ask them so that you don't accidentally add them, and write a 5-page introduction. When I say intro should be a maximum of 1.5 pages, why is it 5 pages? I don't get it. I don't understand. When I say people shouldn't be in the method section, why do I see Dr. B did this, Dr. B did that? I don't understand. So please, I'm doing this for you. Please make sure you take good notes so that you can earn the maximum number of points possible. Okay. So what else have we talked about over the course of the 3 weeks? We've talked about background information, and I can even go back and show us some of that background information from week 7. Okay, so you have this stuff. It's in a video from week 7, and we wrote notes on it, and it's here for you. So these are the things that should probably be incorporated into very organized, logically flowing paragraphs. Concise, small. Okay. So if we go back to what we're doing today, not only that, but during our 3 weeks of talking about things and how things work, we also talked about certain methods. We have discussed that for this report, because there's several techniques that have to be done, this method section is going to be broken down into subsections. Versus the TLC report, it was just TLC, and that is it. There was no individual techniques. From start to finish, it was TLC. This one, we have certain subsections Those subsections have titles. We have definitely written down relevant details that belong in those subsections and have crossed out things that have not. Word economy, everyone. That means using the fewest number of words to get the same point across. So things like telling a researcher how to make solutions, like telling them add 50 microliters of this, stir it on a stir plate, things like that. Making solutions, labeling tubes, that is math that a scientist can do. The minimum details that a scientist needs is the final volume of the whole solution that's being made and final concentrations. With that information, a scientist can make the exact same thing. So we've talked about how to do this. And that was last week. So here we go. Not there. Talked about a lot of stuff last week. So these are some of the things, and I even highlighted them for you with big, giant bullet points. So take that into consideration when you're writing your final draft. Okay. Let's see. Transferring the nitrocellulose, big, giant bullet points. We've basically done this. Just turn it into a report. Method section. Okay. So what are we going to do today? We are going to talk about the results for the Ponceau stain, and we are not going to talk about the results and the discussion for the immunoblot, because the approach that we use for the Ponceau stain can very similarly be applied to the results and the discussion of the immunoblot. So this is kind of like, hey, we're going to do it together for one set of data. Take good notes, think about the overall approach, and then use that for the other set of data on your own with your team. Okay. And don't forget that results, we've also already discussed the Bradford assay, the protein concentration. I've gone through outlines 1 and outlines 2 and provided teams with feedback. I've reminded you to look at feedback from the first time you did a serial dilution back in the beginning of the semester. So all of the feedback cumulatively is expected to be applied moving forward, always. Okay? So if, for example, in the Bradford assay, if I told you don't use graphical analysis, that feedback also applies now. Don't use graphical analysis. That app is trash. Okay, it doesn't make formal figures. And I've showed you different ways to make formal figures. So I expect that formal figures are made. If I said, hey, in the past, your captions are missing relevant details, or captions are not the place to explain the data, fix it. Don't do that again. If I said, yeah, your data points need values next to them and they need to be described in the caption, do that. Okay, so this report is worth 75 points. So it's a bigger part of your lab report points. Okay, so here's what's going to happen. You are going to be broken into small breakout rooms, 2 to 3 people, which means you definitely have to participate. I'm going to even try to make it 2, but depends on the number of people that are here. Oh, no, because I'm one of the people. So it's going to be groups of 2, maybe a team of 3, but everyone needs to contribute. What you're going to do is start a Google Doc. You're going to share that with everyone. I'll pick the person in just a second. And everyone's going to be contributing to this document. When we come back from breakout room, the stuff that we discuss as a class, maybe put it in a different color font so you can see the difference in notes, or start a different page or whatever you want, so that you have a very good set of notes to take with you to start writing your report. No, we're not doing this. You're not submitting to Canvas by 2 a.m. or p.m. Okay, you're not submitting it to Canvas. But it's in your best interest to take really good notes. Okay, so you don't have to convert it to a PDF. You just have to make a Google Doc, share it with your breakout room. What you should have out with you is your outline number 3, okay? Probably also of interest, and that will be helpful for breakout sessions, is probably the Western Law Manual, and then also the report expectations. So these 3 things probably go hand in hand, but I think most of it will be gleaned from your outline that you turn in. And you may or may not be with people you wrote the outline with. That part doesn't matter. It's about sharing ideas or confirming ideas if you are with your writing partner. Okay, so here's what you're going to do in your next breakout room. And I'm going to change some of this stuff after what I've learned from lab yesterday. Okay, so definitely what you're going to do is, I think it would be a nice idea to separate your document. Maybe you can have just make one giant table with 2 columns, and the title of one column is going to be results, right? And the title of the other column is going to be discussion. Okay. And so the reason why we're doing this side by side is because when you write your results section, you should, when you write your results section, you're going to be picking samples to describe first, and then second, and then third, right? That manner that you chose for your results section should be the same order in which you discuss, means explain the data. What does the data mean? Why did you get the data? You are going to discuss them in the same order. So that means both sections are consistent with one another. Okay? And so you're not all over the place. You're not, using all these different organizations for different sections. It's very clearly paralleled. They're almost mirror images of one another, but not really, because one of them, you're just stating the data, pointing out specific things, and you're basically planting seeds. The discussion, you're going to explain more about that. Okay, so look at this. Okay, let's see. What you want to do is, for the results section, you want to you can copy and paste this from your outline, and people should have topic sentences and transitions in their outline because that's part of a detailed outline. So I was a little disappointed that group said that they didn't have a transition statement, or they didn't have time to come up with one, because you definitely should have, because that was due in the homework. So you're going to take the transition statement from the previous paragraph, and that previous paragraph, to just jog your memory, was about the protein concentrations of the plant lysates. Right? So that transition sentence should have something to do with maybe what happens next after taking the protein concentration. What are the things that happen next? And then you have your next paragraph start with a topic sentence. So this is your topic sentence about Ponceau. Okay. And then you start talking about the Ponceau results. So one of the things that you're going to do in your breakout room, here's what we're going to say. This is, I think, is going to be better organization for you. What are the similar data or trends, whatever you want to refer to them as, for all the samples? What is common for all of the samples? Okay, you just need a bullet point list. Okay. Then you talk about your samples one at a time. So I'm going to ask you, before we go to a breakout room, tell me, put it in the chat, tell me what sample you talked about first. It's not a trick question. Just want to know what sample was described first, the data from which sample. Okay. Great. So most of you are saying the plant that was grown in a 12-hour photo period. So now I would like for you to very briefly, why did you talk about that? Even if you just like randomly did it, but what was your reason for talking about that first? Why? Absolutely. So people that are answering, most of you are saying something along the lines of, it is the first sample from left to right if you're looking at the Ponceau stain. And that's exactly what you want. It's not because it's the positive control. It's not because anything else. It's because of the way it appears on the figure. So the way that you read words. So if you're reading this line here, you read from left to right. This is the same way you should read figures. This is the same way you should read tables. This is the same way tables should be described. They should be described from left to right, then top to bottom. If you're reading a piece of paper, first you start at the top, left, left to right, and then you move to the bottom, left to right. And that's the way that you should describe them. So not only are your results going to be consistent with the way that you present your results in the discussion, they're also going to complement or be consistent with the way that they visually appear on your data. And I think that that is the most consistent way to do it, and it keeps things very organized. So if positive control was the first thing that you see, that is the first control that you talk about. And in general, when you're the person that's running the experiment, you should keep that in mind. You should put your controls first, because those are the things that are going to tell the scientist, you or your reader, hey, this experiment actually has some validity because the controls worked. Cool. So you should, when you're writing about it, you should want to convince your reader of that first. Like, all right, and before you start trying to question me, the controls are working perfectly fine. So you have your positive control. So what is the data that is only specific to the positive control? Okay. Because you've said all the other stuff. There's no need to repeat all of the data that you just said was similar for all of the samples. That is the definition of being redundant. Okay? And that is a way that you can write more concisely. Or this is a way. All right. So having said that, and just with the information that I gave you, if you need to talk about your positive controls first, hopefully the scientists that ran the experiment put the next control. Then I did. So you write about the next control. What is the data that is specific to only the negative control? Not only that, compare it to the other control. That's another thing I'm seeing lack of in the TLC reports. It just reads like a bunch of data. The RF value for this was that. The RF value for this was that. Okay, great. The RF value range for the polar amino acids was this. The RF range for the non-polar was this. Compare them to one another. It was less than the polar amino acids. Surprising. You can use short descriptive phrases or even just like words. Similarly, the negative or plants grown in the absence of light, blah, 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 blah. That still keeps it concise, but it adds a little character to your report. And it doesn't sound like a robot is just spitting out data. All right? Okay. Take that out of it. All right. Then what's left? The actual sample or samples of interest. We are only testing one variable at one variation. So we're testing light duration, true, that's the variable we're testing, but we're only testing one type of that. We're only testing that variable at 60 minutes of light. So what is the data that is specific to this sample, and then you compare it to both controls. And keep it in the same order. So compare it to the positive control first, compare it to the negative control second. We're keeping everything in the same order every single time from start to finish. Now, once you're done describing all of the information, you want to use actual specific data. So when you come back from your breakout room, here's what you're not going to tell me. If I said, hey, group 3, what did you talk about for your positive control? I do not want to hear, we talked about the data that was specific to the positive control. Cool. What is the data that is specific to the positive control? That is what you need to report back to class, the actual specific data. Okay, I'm not asking you to just repeat this to me. I'm asking for the actual specific data. So when you leave here, you have almost the exact information that you need to write your report. At the end of this paragraph, you need a transition statement, sentence, whatever. And that transition sentence needs to take you from briefly from talking about Ponceau to talking about immunoblotting. And if that is the end of your sentence, then you should have the topic sentence for the next paragraph should be about the immunoblot only. And those two sentences have to be different enough so it doesn't sound like you're saying the same thing twice. But let's call this part, like one thing that you can kind of maybe consider is don't talk about too many details. So just like a general. And then here, maybe you can put a tiny little bit more details as to what the immunoblot, something about the actual immunoblot that you're doing. The transition statement just kind of like drops it like, hey, next we're talking about immunoblotting. Okay. All right, so that's what you're going to do for the results section. And then the discussion section, here's how we're going to do this part. I'm going to try and move this down. Okay. So you're going to keep using the same, whatever it's called, order. You're going to keep being consistent. But discussion, what are the things that go in a discussion? Discussions require your expectations. So here's where you say, we expect if Ponceau stain worked properly, we expect blah, blah, blah to happen. But not only that, we're talking 1 to 2 sentences. Let's keep it red. Ponceau's red. What's the mechanism? We're talking 1 to 2 sentences. And maybe that's part of your topic sentence. I don't know. Okay. Because that is what kind of provides the rationale for your expectation. So this kind of leads you to what you expect. Okay. So if it wasn't clear, because it wasn't, no. Oh, shh. Okay. Okay, so you want your transition sentence from the previous paragraph. So in the discussion, you have already discussed your concentration of your protein lysates. So how do we get from that idea to the discussion paragraph for Ponceau? So you want your transition sentence and your topic sentence for the Ponceau discussion. So this is for Ponceau. Okay. This paragraph, or keep in mind that the discussion could be a couple different paragraphs. So the Ponceau stain mechanism is what helps you rationalize your expectations. Ponceau stain, blah, blah, blah. Thus, we expected ABC. Okay. From there, you can start talking and explaining and discussing and speculating about the data. So you start with the positive control, just like you did before. And then you wanna, if there's anything specific, so what do you expect in general? So I know you know this one. So what does Ponceau do? What is like, if you had to do it in a phrase, what does Ponceau do? Tell me in the chat. Not what does it show you, but what is the mechanism? Like, what is the molecule of Ponceau doing? And maybe you have a little more information about that. So Ponceau stains proteins non-specifically, and maybe you want to just like tell me how, right? So there is what part of the protein is a bind. Okay, cool. Right? So I know you know. We're not putting it on the sheet, but I know you know. Ponceau stain works, blah, blah, blah. Thus, we expect, so tell me, what do you expect to see in general? Not specifically, just in general. No matter what sample you were testing, what would you expect to see if Ponceau was working? Describe what the data would look like to you. Okay, what would you expect the Ponceau stain to look like? Yeah, what would you expect the Ponceau stain to look like if you had to describe the whole entire membrane? Let's pretend you had 10 samples. What would you expect to see for all 10 samples? Okay. Okay, so a lot of you are still understanding what Ponceau does, and you can properly describe what you should actually see with your eyeballs. And so, yeah, if Ponceau worked, you would assume that no matter what tissue or what cell you extracted protein from, that cell is going to make proteins of all different sizes, meaning all different kilodaltons, all different lengths of amino acid chains. Maybe some cells make a lot of protein that are 50 kilodaltons big. Maybe other cells make proteins that are 20 kilodaltons big. So the point is, cells are going to make proteins of all different sizes in all different amounts. So if Ponceau binds to all of them, then what you should see, like you're saying, is you're going to see a variation of red bands, or you can say red bands, and I think of varying thicknesses, of varying lengths, of varying kilodaltons. Yeah, red bands, different sizes, different amounts. Yeah, okay. That's in general. Okay. And then why? We just said it, because organisms, tissues, cells make proteins of those same characteristics, of different sizes and of differing amounts. There's your rationale. That is how you properly rationalize your expectation. So you're telling the reader what you expect to see in terms of the data. The data are red bands. The data is more or less red bands. The data is red bands that span from 10 kilodaltons to 250 kilodaltons. The rationalization, or the rationale for that, is that since Ponceau non-specifically binds to proteins, all cells or tissues or organisms are going to make proteins of all different amounts and sizes. That is your rationale for the expectation of your data. That is how you properly discuss data. I'm seeing in a TLC report that people are just describing expectations with no rationale, with no references, with no nothing, and then the discussion's over. The discussion is the bulk of your points. So when the discussion is only 2 pages and the introduction is 5, there's an issue. And that's the amount of effort that should be placed on certain sections is reflected by the point value. So check it out. Okay, Do we not mention Rubisco at all? There is an option too, but don't put too much attention to Rubisco. Why? Because Ponceau stains all proteins. And we're not talking about all proteins if we're just pointing out Rubisco. I think Rubisco can come into play a little bit. I don't know if it would be a good transition sentence. It depends what the transition is and the topic for the next paragraph is. So I can't answer that. So once you describe your expectation, generally, just like we did here, we described this stuff generally. Now you can move into your positive control. What did you specifically expect to see? This is when maybe you want to bring in Rubisco, mention that possibly we might see a couple very thick bands and blah, blah, blah. This might be a good place. And then you need to describe why. Why did you expect to see those specific data? And then recap the trends that you did see. That should be up here too, actually. Move this down to a little more space. The question is, did the data support this, yes or no? And one way to do it is just say, as expected, or expectedly, or with no surprise. That's kind of a weird one, but the data, in fact, showed that Ponceau worked properly, or however you want to say it. If the data did not show that, that's going to require a little bit more work, because then you have to describe what the data did show and why that happened. So it's easy. If the data did support the expectation, then you need an overall conclusion sentence for that. Or you can even kind of combine them into one sentence. Okay. I need to keep moving this stuff. Move. Okay. So I'll ask you, if the Ponceau did work properly, what conclusions can you make? It could be about the progress of the experiment. So I'll just give you some ideas. Progress of the experiment. Okay. So that's of the actual methodology. But then you want to also consider, so if I asked you, don't put in the chat, but I know that you, because we talked about this, like, hey, if you did see red banding in all the lanes, what has worked so far? And you would be able to tell me a few things. And if I said, what about the data shows you that, I'll give you one example. What about the data shows you that the transfer was successful? You would be able to say, oh, well, because I see red bands on the nitrocellulose. Boom, that's all we're looking for. That is a conclusion. That is concluding what the data means. Okay? So not only can you talk about the progress of the experiment, but you can also talk about, what is the thing that we're doing here? The variable. What is the variable? Duration of light. So you need to make some general conclusion. Let's keep that one to the end. Put it on pause. Okay. When you're talking about your positive control, what do you expect to specifically see? Maybe you start dropping hints about Rubisco here. Why do you expect to see those specific data? Did you see those specific data? What is the conclusion that you can make? So if the variable is the duration of light, what can we say about a 12-hour photo period? And the ability for plants to synthesize proteins, right? Because that's what we're looking at. We're just looking at the plant's ability to make a bunch of proteins of different sizes. So this is more about protein synthesis. And then you make a maybe conclusion. The data also may suggest something about Rubisco expression. And if you don't have the word may or possible or potential, you're going to get points off, because Ponceau does not tell you specifically with 100% confidence anything about Rubisco. You don't have that confidence. You could be like pretty sure, but not 100% sure, because Ponceau is a non-specific protein binder. Let's see. Is it okay to justify our expectations with what we saw in the Bradford assay? Yes, that is key. That is taking sets of data and not comparing them to one another per se, but really pointing out that they support one another. So that is evidence of somebody who's really deeply critically thinking about the data sets as a whole, and not just individual samples, not just individual data sets, but the entire study. That is excellent. So if you're doing that, do it. If you haven't considered it, consider it. That is the difference between an A and probably not an A. Okay, more in the absence of light because we saw higher concentrations. Sure, so that's kind of like a team specific question. So I'm just gonna generally answer the question, yes, sets of data should be compared or should be talked about in terms of do they support one another. I think that's great. It's going to be most important. So if you're not paying attention, pay attention now. It's going to be most important to make a relationship or identify or point out similarities between the Ponceau stain and the immunoblot results. If you don't do that, you are missing the bulk of this experiment. Arlene.
Sorry, I thought I unmuted myself. That would fit into the progress of the experiment, right? Where general conclusions are kind of going.
You could probably fit it there. Yeah.
Okay.
So, yeah, it depends on how you write, but I can see it fitting there. So if the conclusion is, so seeing the bands in all three samples, this indicates that blah, 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 and that protein lysate was, whatever. If you want, I don't want to like blow it for everyone, like give everyone the answer, but yes, you're on the.
Okay. Thank you.
You're welcome. Okay, cool. So next, since you've already done this for the positive control, then you might want to move to the negative control. But you do the same exact thing. Okay, so because we're doing the same exact thing, you got them away. We are going to take this. We are going to put it there. Okay. For the negative control, what do you specifically expect to see? Do we want to mention anything about those special regions that may have a kind of planting some seeds about Rubisco? Why do you expect to see that? Does the data show that? What does that suggest about the 12-hour photo here? Not 12-hour. Absence of light. Okay. Now the discussion, this already is much bigger, right? There's much more information here, which is what the discussion should be. Results should be one paragraph. To describe one paragraph worth of results, probably going to be 2, maybe even 3 paragraphs, depending on how concise of a writer you are. So let me provide you with some ideas of how you can organize your paragraphs. So you can potentially, and we now know that anytime you change paragraphs, there definitely should be a transition sentence and then a topic sentence for the next paragraph. So maybe your Ponceau paragraph, the first one, nice and short, but it's just about the general statement and what you can generally understand from Ponceau. Then maybe your controls go in the next paragraph. Okay, so maybe the next paragraph is just establishing your expectations for controls and what they mean and what they show you. Okay. All right. Oh, one thing that we forgot to do here that we have to keep consistent with the results. Compare this data, just a quick reminder, to the positive control data. And that could be Unlike the positive control, it could be as simple as that. Unlike the positive control, plants grown in the absence of light had a very thin band at blah, blah, blah. Okay, that's the specific, that's it. That's comparing it. Great, you did it. And it points that important thing out for the reader. Like, oh, wow, yeah, that is a positive and a very clear negative control. Wow, these people are geniuses. Okay. All right. So maybe the last paragraph, and I would be surprised if you can squish this into 2 paragraphs. I'm probably going to suggest you do 3 unless you blow me away. That's possible too. But the last one is going to be your testable sample. Testable sample. Okay. But you're going to do the same thing. It is a template. Science writing is this boring template. You just kind of have to switch the words around a little bit and the order around a little bit just so that it doesn't sound so boring. Okay, but it's the same exact stuff. Okay. Oh man. Let's do it again. Copy. Right. And this time it's not the absence of light, but it's 60 minutes of light. Not only do you want to compare it to the positive control and do that first, you also want to compare it to the negative control. So if you didn't put this much thought and energy into the discussion for just the Ponceau data, that's okay because it's not the final draft yet, but this is the type of thought that needs to go into the big home run data sets for this study. That's the Ponceau and the immune blot. The Bradford, I mean, spend some time in it, but like not too much because that's just making the agents, right? You might just bring a couple different points up, but this is the amount of effort that goes into discussing data properly. Okay. Last but not least, when you're done with this, you are going to write your final transition sentence that is going to take you from talking about, not stating the results, talking about, discussing, explaining the results of the Ponceau stain, and you want to somehow create a transition statement that takes you from Ponceau explanations to, hey, now we're going to start talking about immunoblot explanations, and then your next topic sentence. So as you can see here, everything is the same, but I think it's really, really obvious the amount of effort that goes into either section. Look how small this information is, which means your results section should be very concise, very tight, very simple and straightforward. You're just stating the results that I should look at with my eyeballs. Another thing that I'm noticing is that people are forgetting to reference their figures. So for example, at the end of talking about your controls, you probably should reference, even if it's like every 2 sentences, once you state some sort of data, at the end of that sentence, you should have figure whatever. This is actual data, so it should be figure 2B, not figure 2A, because 2A should have been your predicted model, but figure 2B. Once you talk about this, figure 2B. Mostly I'm seeing it absent in the discussion. If you're talking about expectation, let me use a different book. If you're talking about expectations, that is the whole point that you made that model. At the end of this, figure 2A. Talk about the data, figure 2B. And you go through every single time you expect something, reference your model. Every time you recap the data, reference your actual data. Anytime you're explaining your rationale, reference an article. So this, you still need to, discussion, you reference your figures and tables, and then you have in-text references. Those are key. I'm seeing a lot of discussions with no references at all. So let's make this Western blot report better. Okay, so it's a lot of stuff to talk about, but it's not new. It's not like you don't have this information. It's not like we haven't talked about it before many times, 3 different weeks. So you have this information, but I realize that you're going to need to, kind of, or maybe reorganize some of your stuff. So I think 25 minutes was a little bit not enough time for the class on Tuesday. I'm providing you with way more direction so that we can come back and knock this out of the park. So I'm going to give you 30 minutes, but that means those 30 minutes, I want you to use all 30 of those minutes. I don't think anyone's going to be done before 20 minutes. So if you're done, you don't have enough information. I'm telling you that right now. So go back, work for the rest of those 10 minutes so that you are fully working for 30 minutes. Okay, so let's call it 20 minutes. It's 10:50 now, plus 30 is 80, 11:20. We're going to be back in class at 11:20. Let's get you into breakout rooms. Okay. Okay, everyone, I hope that was enough time. So we'll go ahead and get started. And again, just a reminder, the quicker that you participate in the chat when you're called upon, and the quicker you contribute to verbally, then the more efficient we'll be, and the quicker we'll get out of here so that you can be on with the rest of your day. So what I thought might be helpful is I copied and pasted the Ponceau stain onto the document here so we can all kind of reference it. So let's go ahead and start with, let's just go with, how many groups were there today? 8. Okay, let's start with group number 7. So I'm going to have you tell us what your transition from the protein lysate concentration to Ponceau was, as well as the topic sentence. And so I am going to be taking notes given what you tell me, but I'm only going to be jotting down certain phrases so that not everyone is going to have the exact same transition sentence. So everyone, you should also be taking notes and not trying to write down word for word. Okay, so group 7, go ahead, unmute yourself and tell us what you said your transition sentence was from concentration, determining concentration to the Ponceau.
Let's see. Apparently, I didn't write that down when I transferred that over. So I literally just missed that. I started with the topic sentence, which we put like the
Okay.
overall
That's
goal.
okay. That's okay.
Overall goal of the study is to see how the exposure to the 60-minute light affects reversible concentration in the plant lead.
But I think it makes most sense to have the pair. So we'll come back to group 7 a little bit later. So let's go ahead and do group 5. Group 5, what's the transition sentence? And then also tell me the topic sentence. Okay, one more time, that's group 5. So Christian or Nancy, please unmute yourself and tell us what the transition and topic sentences are.
All right, my bad, I'm sorry. Transition is to investigate how Rubisco protein gene expression would be expressed in P sativum after being exposed to a 60-minute photo period.
So that sounds like the objective. That doesn't sound like you just finished talking about protein concentration.
Okay.
So how do you go from talking about, so we're in the results. How do you go from talking about protein concentration to now talking about Ponceau staining?
Sorry, give me a quick second. Can you repeat the question?
Yes. So a transition sentence takes you from the end of a paragraph to the start of the next paragraph. The previous paragraph, you just finished talking about how you determined the protein concentration of your samples to be this, this, and this. The next paragraph is going to be about Ponceau staining results. So how do you connect? How do you go from protein concentration to Ponceau?
Sorry, I'm gonna ask my partner to answer that.
It's okay. It's okay. We'll come back to you. We'll come back to group 5. So maybe you'll be ready to share something from a different part of this. I really don't want to wait around. I want to make sure you guys get out as soon as possible because I know that there's an exam tomorrow. So let's go with
Very cool.
room 3. Please unmute yourself and tell us what you have.
Yeah, so our transition was once concentrations are calculated, the proteins must be separated into different sections by weight, transferred and then transferred and stained to ensure the existence of such protein concentrations. And then our topic sentence was a Ponceau stain allowed us to visualize the success of the Bradford assay and transfer to the nitrocellulose membrane to verify presence of samples.
Awesome job, group 3. Okay. So I generalized it because I want groups to be able to put their own spin on it. But to recap what Avery and Greg discussed was that once they were done talking about their protein concentrations for the plants grown in a 12-hour photo period, yada, 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 they said once concentrations were determined, that's what this thing is right here. Once the protein concentrations were determined, then they basically just listed the steps that happened in chronological order. They said these lysates were separated by size using, I would probably say, using SDS page, comma. So these lysates, were separated by size using SDS page, then transferred to a nitrocellulose membrane for further analysis. Further analysis, further investigation, further observation, whatever, further science. Keep it simple. There was a lot going on in that transition sentence. I think that you can trim it down. And now the reader knows that you've done some stuff. You've run the stuff through a gel, you've transferred to a nitrocellulose membrane, and so now you're just allowing them to progress through time. And that naturally leads you to the topic sentence. And these two sentences pair well together. That doesn't mean these are the only possible combinations, but it is a possible combination, right? So the next paragraph starts with the topic sentence, and now we're going to get a little more specific about what this paragraph is all about, and we know it's about Ponceau results. So once proteins were transferred to a nitrocellulose membrane, Ponceau was performed too, and then this group said they briefly just reminded what Ponceau does to ensure blah, blah, blah. I think that is great, and this is the exact approach that I want to see in the reports. Okay, so after NC, then Ponceau, to ABC. Great job, thank you. So we're going to go back to group 7. Thank you, group 3. Group 7. Go ahead and tell us some of the similar data. Let me see if I can make this smaller so we can all see the supposed. I'm going to move this for now. Okay. Let's talk about the similar data. Group 7, go ahead and unmute yourself. Tell us any and all of the similar data that you can say is common between all 3 samples. Oh, before we get going, I think you can clearly see that there's 2 lanes of, what are these called? Molecular weight markers. And I think in the video I said like it bled over into the next well. Okay, so here's what you can do. You can definitely just crop this picture so that it only has 1 lane of the molecular weight marker. If you don't want to crop it, so whatever is easiest for you. If you don't want to crop it, you can just say that both lanes in 1 and 2 are, and you just say, you know, you provide the official product name. And there's no need to explain why or anything like that. This is not data. This is not sample that you're interested in. It's your molecular weight marker. And that also just shows you that science happened in real life, even for me, some supposed professional. And so I needed to make a decision on the spot. And I could see the marker bleed into the next well. And I was like, well, I'm definitely not putting my sample in this well as well. If I did, you would have had this banding pattern and this banding pattern in one lane, and that would not have been observable. Like you wouldn't be able to make conclusions about it. Okay. So, question after group 7. Okay, group 7, similar data. Hit it.
Bands of varying sizes throughout their entire molecular weight ranges, and then
Can
all
you
of
be
them
more
have-
specific? So what is the range?
specifically, my group looked it up, and we saw like very specifically bands ranging for positive from 10 to 75, for negative bands ranging from 10 to 100 kD, and then from the 60-minute bands ranging from 10 to 150 kD.
Okay. But in general, what's similar between them is at least 10 to 75, right?
Yes. Mhm.
Okay, so you can specify that and you can put that, and that is common for all of the samples. So of varying sizes.
Okay, and then all of them have thicker, slightly thicker banding around 13 and 55 kD.
Okay. I see that. I would. Excuse me. No. There's no one. Scare the crap out of me. Stop. Okay. Yeah. So slightly or somewhat thicker, right? Bands at approximately these specific sizes. Great. And so I just want to draw everyone's attention to how you can use a critical eye. So everyone is saying, well, I see there's a big, huge fat band here. However, look at the molecular, this is why molecular markers are so important. You can see the molecular marker here not only has this orangish color band, but also a blue band smushed right above it. I could have copied and pasted the molecular marker info sheet, but essentially when you are running your Western blot, depending on the percent of acrylamide, a higher percent is going to allow smaller things to resolve from one another. A lower percent means the smaller things are going to basically separate. They're not going to separate from one another and they're going to run with one another. So you can even see that the percentage gel that we used didn't even allow the molecular marker bands to separate from one another. So even though people are saying that they see thicker bands there, which is true, that's an observation, you got to think about what that actually could be. Don't just assume that that's the small subunit. What you should start thinking like, oh, well, maybe that's just all of the small proteins that the plant is making. They didn't resolve from one another. What's the evidence for that? When I compare it to the molecular marker, the smallest proteins in the molecular marker also did not resolve or separate from one another. Just keep it in your mind. So make sure that you're not jumping to inaccurate conclusions. Because if you said this must mean that that's the 10K, that's the small subunit of Rubisco, your data does not indicate that. Your data indicates that the smallest proteins did not resolve from them. So be careful with the conclusions you make. Okay. Anything else that's similar for all three sets?
I mean, I guess a band near 37 kD. And
Okay,
another,
great.
but
Great that you mentioned that, but now the question is, does that play a role in this study? Yes or no?
no.
Okay, so the stuff that we wrote here are the things that are probably important for the conclusions we're going to make. So this is definitely important for some conclusion we're going to make in the discussion.
Mhm.
This is definitely important for conclusions we're going to make in the discussion, as well as something that we can compare to for Rubisco later. So these, I would say, that's it. We're good. This is done. That's the only thing that's similar. Okay, great. And go ahead, Megan, what's your question?
It was regarding back to the topic sentence for the Ponceau
Okay.
staining. So I just want to clarify, we talk about, so instead of having the nitrocellulose membrane transferring in the transition, we put that in our topic sentence?
Hmm.
I'm just trying to, it says after NC, then Ponceau.
Yeah, so you're kind of like connecting it. So here you're saying that the proteins were separated by size in an SPS page and then transferred to nitrocellulose for further investigation. So let's just put this in parentheses, right? Because it really just depends on your writing. So if you didn't add that in the previous statement, you can start the next paragraph. After SDS page, proteins were transferred to a nitrocellulose membrane for Ponceau staining, something like that. But essentially what you want to make sure is that you don't skip any major steps. You can't just go from proteins in a tube to Ponceau staining on a membrane. How did you get there? So what are the details that have to be filled in? And so again, it's really just up to you and your team's writing.
Okay, thank you.
Yeah. Okay, so we're back to group 5. We're going to specifically, so what did we say we needed to do? We needed to talk about the specific data that is only visible or only applies to the positive control. So there's no, I just copied and pasted this image, so there's no labels on it, but you know, at this point, you definitely know that you should probably have some of the important KDs next to these bands, not over here, literally next to the bands on the gel, especially if you cut it off here. Okay, then you can have it right on the edge. You need labels up here. Access other published work to see how simple their labels are on their gel. You don't want to crowd up the gel or crowd up the figure with a bunch of stuff to look at. Keep the abbreviations simple up here and then use the caption. That's what a caption's for, is so that you can use abbreviations and then provide information about those abbreviations in the caption. Okay, crop out whatever you don't need, and then you go from there. So group 5, what are some of the data that are specific to the positive control only that have not been already discussed in the previous sentence?
So for the positive control group, we approximated around 10 red bands, and with the thickest being around the range of 50 KD to 70, around 50 KD and 10 KD. Kilodaltons, I'm sorry.
That's okay. Okay, so I'm going to ask you a question. Think about the overall goal of the study. Do you think that specifying the exact number of bands is something that you're going to use later?
Yes.
So you use the number 10, like, so you say there's 10 specific bands. So 10 specific bands has something to do with Rubisco?
Oh, no, I'm sorry. I thought you were talking about the 2 highest concentrations, but I wouldn't say the number of bands counts, but we can say that when we notice in the other groups, we can be like, oh, the plant experiences different reactions that require different enzymes that are not Rubisco. Something like that.
You can't say that here because you don't know what proteins are there or not, but save that for the discussion because it could be something that's important later.
Cool.
But what can you say about this? Let's just keep it simple. So the only piece of data that applies to the positive control is that, besides what we've already talked about, is that the thickest band appears at approximately 50 KD. So you can use these phrases. Approximately 50 KD, which indicates you must have known that this band here
is Rubisco.
Was 50, not Rubisco. It's a protein that is 50 kilodaltons in length. And this one is slightly higher, right? Slightly larger. So that means it's slightly larger than 50, or you can say approximately 50. If something was slightly below, like this is slightly below, so you would say slightly smaller than. So when you're describing bands, smaller and larger point at KD. So a smaller band means a smaller number. So not 50, but like 45 KD. Smaller does not mean thinner. So everyone needs to get that straight. If you're going to be describing the intensity or the thickness of bands, you need to use those two phrases. More intense, less intense, more bold, less bold. That's a weird one. Thicker, thinner. Those are used to describe how fat the band is. Higher or lower on a gel, we don't say those words. We don't say higher or lower. We say larger than, smaller than. Okay. All right. So that's the only thing that we can say about the positive control. I know that you have already described earlier what the positive control is, but you have to understand that the sections are kind of like their own mini reports. So the first time you start talking about these samples, you need to describe them one more time. So group 5, tell me, what is the positive control?
It is a group of pea plants that experience a 12-hour photo period.
He's grown in, wow. A 12-hour photo period. And you're going to write all these words out. I'm just taking notes right now. So peas that were grown in a 12-hour photo period, and then in parentheses, you can actually have the phrase positive control, not the symbol like I wrote it, not plus con, none of that. We'll write positive control. Displayed a very thick band at approximately this and this kb period. Figure 2a, whatever it is, right? Whatever figure. So you would have, don't forget to reference whatever figure it is at the end of the sentence. And here you'll do the same thing. Okay, great. Thank you. Let's stay with this group and let's do the negative control. So let's start with, what is the negative control?
The negative control group is a group of pea plants that experience no light exposure.
Okay.
And they expressed thick bands at 10 kilodaltons and a range between 50 kilodaltons to 70 kilodaltons.
Okay. So again, the range, didn't we already talk about the range up here? All of the samples had that range?
Yeah.
So saying it again, redundant. Take it off.
Okay.
Okay, so you can kind of make the difference between these two sentences by saying things like, unlike the positive control, or dissimilarly, or conversely. So some kind of word here that allows the reader to know that you're going to see different results. Okay, so if you are starting with like, oh well, these the negative control is also displayed a band at approximately 10 kD. Okay, so that's kind of similar to the positive control. Plants grown in the absence of light also displayed a band at approximately 10 kD. But this goes back to what I just told you guys a couple minutes ago. Are you pointing that out because it's the thickest band and that's the only thing you're thinking about? Or could it be that it's thick for all of them because that's all of the samples smallest, like just all the chunk of all the proteins that are small sized for positive plants, negative plants, 60-minute plants, for all of the plants, these are all of the small proteins. Is that what it is? And if that is what you think it is, then is it worth keep mentioning that there's a thick band at 10 kD when you've already said it up here as well? I don't think you need to keep pointing it out. But when you get to the discussion, you definitely need to say why you don't think that's the small subunit.
Gotcha.
While all samples display a band at the size of the Rubisco small subunit, this is likely not representative of the small subunit, because then that's when you talk about how you use the molecular weight marker to say, like, it's probably just all of the random small proteins that did not resolve with one another, from one another, because of the percentage of gel. So go ahead.
So we just see that we don't see a, like in contrast to the positive group, the pea plants that experienced no light exposure did not see a thick band at 50 kilodaltons.
Yes. So peas grown in absence of light, dissimilar or conversely or unlike peas grown in the 12-hour photoperiod. Lack of the band at 50 was much thinner. So basically you're comparing it to. So much thinner, less intense, whatever you want to say, compared. Somehow you're going to write it so that the reader knows that you're specifically comparing it to the positive control. Okay. So I'm going to just highlight this one more time, because you really want to make sure that in the discussion, you bring up that this is likely not the small subunit. Likely. And then the evidence for that is the molecular weight marker. Keep it in mind. Because otherwise, how are you going to describe that the band here is much thinner? So and here it's thicker. So what are you saying? Like, okay, when plants are not grown in light, somehow they only make the small subunit and lots of it. That doesn't make sense. And that's going to be really hard to explain. And when they're not grown in light, they only make the small, but somehow they don't make the large subunit. And it's not logical. It doesn't seem normal. So be careful with what you point out. That's an example of using a critical eye, seeing this, but then using critical thinking to really think about what that would mean. Okay, great. Thank you, group 5. Thank you, Christian, for sharing. Let's move this down. Okay, last but not least, so we did 7, we did 5. Let's go back to, we did 3. Let's go room 1. Room 1. What is the data that has not been already described up here that is different and unique for our MVPs? This is like who we care more, our superstar. Okay, grown in a 60-minute light exposure. Okay, so what's the data that we should put down on these notes?
Data that hasn't already been discussed. Sorry, one second. I'm just looking over what we've written so far. I'm going to be completely honest. Basically, what we have down is what we've got written.
Well, then if you don't think you have the right stuff, here is the figure. Look at it. What is unique about this versus the other samples?
Well, it's a lot thicker.
What's a lot thicker?
The experimental sample, the bands are thicker and darker than
Okay.
the other two.
Okay.
controls.
Okay. So it seems like overall there's more banding or more red bands or a greater intensity of red pixelation, however you want to say it, but we'll just keep it pretty simple for the notes right here. More bands compared to both controls, right?
Mhm.
Okay. And what is at least the one of the locations of interest that you probably should talk about?
Looks like the thickest are around 50 kilodaltons.
Okay, but now we need to compare it to the controls because that's kind of where we're going to be drawing conclusions anyways. So
Okay.
compare it to the positive control first. So we're talking about the band at about 50 KT. How do we compare it to the positive control?
Well, to me, it still looks a little thicker, but it's definitely closer resembling the positive control than the negative control, I would say.
So let's
The
do one
negative
at a
is-
time. Let's just do
Okay,
one at a time.
Sure.
Here's the band at 50.
Mhm.
Here, I'm going to even highlight it. How do you compare those two? Compared to the positive control, the band at 50K, at approximately 50K for plants grown in a 60-minute light exposure was what?
Well, I would say that the positive control was thicker.
Okay, but you want to
Mhm.
compare the testable sample
Okay,
to
testable
positive.
sample is thinner than the positive control at 50 KD.
Great. Okay, now compare it to the negative control.
Thickness, pretty close. I would say it's darker than the negative control, but they're about the same thickness as far as I can tell.
Okay. So if that's what you're going to go with, so I would just say be descriptive with your observation. So I'm going to leave it here as come up with a descriptive observation. Because there's a couple things that you said that I think are worthwhile mentioning here. One is that you said the testable sample looks to be like more intense, like it's darker. But you said thickness wise is about the same. So you can use words like slightly. You can use words like more intense. So more red pixels means more of that protein of that size. So I'll just keep it like that. If there's more red there, that means there's more of it there. More proteins of that size present. So we'll just keep it like that. Okay. And that's everything that I asked you guys to do, right? So we covered the similar data or the trends for all the samples. We talked specifically about the positive control. We talked specifically about the negative control and then compared it to the positive control. Maybe in one sentence, you don't have to do one at a time. Like you can definitely compare these. You can definitely combine them in one sentence. Then we talked about the data specific to 60 minutes, as well as somehow comparing it to both controls. It's all there for you right here. And if you look at it in terms of numbers of sentences, this makes a very clean, concise paragraph. There's 1, 2, 3, maybe even, depends if you're a good writer, the controls could all be one sentence, but let's just err on the side of maximum number. 4, 5. What at the end of the sentence what at the end of the paragraph do you need? You need a transition sentence going from Ponceau to immunoblot. Okay. And then that is the end of this paragraph. So let's just draw a little line so that we know that that would be the end of the paragraph. Okay, but we want to pair this with the topic sentence for the next paragraph. So here's what we're going to do. Let's go with group 2. Group 2, you're going to tell me what was the end sentence for this paragraph, and then what was the beginning sentence for the immunoblot results. Group 2.
Our transition sentence was that following the Ponceau stain, in order to get a firmer grasp on the proteins, an immunoblot was performed.
Okay, so after Ponceau, all right, so after Ponceau, immunoblot was done to gain further, more specific, more detailed information.
Yeah, for further
Okay,
information
sure.
on the proteins present.
About all the proteins present?
On Rubisco, I guess, in a sense, because that's what we're focusing on.
Perfecto. What was your topic sentence for the next one?
An immunoblot stain will provide information, or the immunoblot stain, sorry, I lost it within the going back and forth. The immunoblot stain provided imaging or showed a stain in which the alkaline phosphate or the AP substrate was present for Rubisco.
Okay, I think this needs some work because it almost makes it seem like alkaline phosphatase is what allows you to It does. It indirectly allows you to see Rubisco, but AP can be used for a million different Western blots. So what is the one specific reagent that is going to allow you to only look at Rubisco?
Is that the antibodies? Using
Which
the
one?
antibody?
Yes. So now we got to get more specific.
Okay.
So which antibody?
The primary antibodies.
What is, yeah, and the primary antibody is specific to what exactly?
Specific to, sorry. When what we have written is it reacts or precipitates when BCIP and NBT react. That's all we have down.
Okay, so if that, so I think maybe, I bet you your classmates can help out. So we know that we're talking about the primary antibody. That's what specifically allows the scientists to see Rubisco, because it attaches to Rubisco. So where does it attach to Rubisco? So somebody in the chat, please tell me. Okay, so it binds to the antigen. Where's the antigen on Rubisco? Okay. Okay. Yeah. So the primary antibody specifically binds a tiny little antigen that exists in the large subunit. And just to remind everyone, a large antigen is just a short chunk of amino acids in a very specific order. So here I like how we talk generally about, after Ponceau stain, the nitrocellulose membrane was subject to, or an immunoblot was performed on the nitrocellulose membrane, basically to get more information about Rubisco expression. So that is kind of a general phrase. And so I think that kind of falls in line with what I said up here, like keep it kind of general in the transition sentence, and then give it a little more detail in the topic sentence. So I think here, we know we're specifically using a primary antibody. An immunoblot was performed with a primary antibody that was specific to, so that's why we say, I'm writing notes, right? So we know specific to means the symbol is alpha. Specific, I don't know why. Specific to the large subunit of Rubisco. That's it. We don't need to talk about all the other antibodies. This is not a material methods paper. And I think it's pretty common knowledge that whatever primary antibody you pick, there is going to be a succession of other antibodies and reagents that allow you to see the bands. Okay, let's finish that last sentence here. So here's your results paragraph for the Ponceau data. 5 to 6 sentences. Okay, very specifically in a very good order, logical order, controls first, comparing controls to one another, then talking about the testable sample, comparing that to the controls, finishing it up with, hey, we just did this, now we're going to do this. The next paragraph starts with like, here's how we're specifically going to do this, and now let me tell you about the results. Okay, any questions on the results section here? Anything you have in your own outline that you felt you should need? This is your workshop because I know how to write. Not great, but I know how to kind of write. Okay, then we will move on. Okay, so where am I going to move this thing to? For now. Okay, so I hope at this point we know how to write transitions and then topic sentences. So for the discussion, for class discussion right now, we're not going to go over that. So now you have an idea, and now you can use the approach that we just used for the results section. You can do the same thing for the discussion. And the sentences could be pretty similar, but you just tweak them a bit so that they're not exactly the same. Okay, so we'll start with the similar data. All right, similar data. All samples. And regardless of what you have on your notes as a team, you've been adding to those notes. You've been adding to those notes. Here they are on this piece of paper. So adjust what you contribute to class based on what we've already discussed. Okay, this means that you're actively staying engaged. You're adapting on the fly. This is what we want. This is what makes a workshop. Okay, so whatever you have on your paper, maybe it still applies. So let's do it. And just to recall what you need to do here, we're going to start with, let's start with this. The Ponceau stain mechanism is going to lead us to what our expectations are for all the samples. And we kind of already talked about this. So I guess we don't need to, right? Yeah, we've already talked about this. So check that off. But let's write it down here to make sure that we know somewhere in here, we talk about the mechanism. This is what allows you to move directly into your expectations, your general expectations. And then after that, now we are talking about the similar data for all samples. So I think this is a class chat thing. So wait to press enter. I want everyone to type in the chat all the things that the Ponceau can tell us about the progress of the experiment. Everything you can think of, please don't press enter. Go ahead, enter. Okay. Okay, so one of the things, we can work backwards in time, right? So one of the things it can show us if the transfer worked. Okay. All right, Avery is indicating that not only does it show if the proteins transfer to the nitrocellulose, but it also tells us whether SDS page really did in fact separate these things by size. So whether or not SDS page worked the way that it should. Okay. Okay. There's one other thing that it will tell you. So if you see red bands, it means there's protein. So what else did you do right? Teenagers part of SDS page extractions, protein extraction. Okay. So now I'm going to ask you for each one. What is the data that tells you, hey, transfer worked? How do you know that? What are you seeing that makes you say the transfer worked? We don't need to use formal language right here. So what is the data that tells you the transfer worked? Okay, the presence of red on the nitrocellulose membrane. Red on NC. And some of this can go in your report, some of it cannot, but definitely if it is going to go in your report, formalize it. So these are just notes that we're taking. Great. What is the data that shows you SDS page worked? What about the data's like, yup, definitely protein separated by size. Okay, bands from a small KD to a large KD range. Great. So bands of varying KD range. Great. Protein extraction. What tells you that protein extraction occurred properly for all the samples? Red in all of the wells. Yes. So transfer, you can make that claim by seeing any red on the nitrocellulose. Like, you know, the transfer apparatus worked. All right. So you can definitely combine all of these into one brief sentence. So Ponceau stains non-specifically to proteins by binding amino acids. We expect to see bands of all whatever varying KDs and of amounts for all 3 samples, indicating that the transfer and SDS page techniques worked properly and protein was successfully extracted from all plant samples. That is your explanation, or that is your sentence. And then you can say this was expected because, or we expected this because. That's when we talk about how cells and tissues and organisms make proteins of all different sizes. Okay, all 3 lines. Right. Okay, cells, organisms make proteins of all sizes. And now we need to talk about whether or not we saw that. So that brings us to our similar data. We already told the reader that we did see that stuff. Okay? So in fact, the data did show red banding for all samples of varying size. Figure 2b. When you talk about these general expectations, you need to remember at the end of this sentence, I don't have any space over here, but at the end of this sentence, you should be talking about figure whatever A. I don't know the number, okay? But that's your predicted model, right? You should have a model that shows that. So the similar data, okay. So see, we did all the odd groups. We started with 2, 4. You got it kind of easy in the start. So group 4, did the data support this expectation, yes or no?
Yes.
Okay. Supported the expectation. You don't really need to do anything more, but just recap what the data did show. That does not mean copy and paste the stuff again. You can just recap that there were varying protein bands of whatever for all of the samples. And then you have figure 2b or figure whatever b at the end. That's it. Okay. Now, group 4, we've kind of already made the conclusion. So this should show you, like, you don't have to do the conclusion in order at the end. Like, we kind of already said this. We already said the conclusion of the progress. So let's make the conclusion about about varying light as the variable of light duration. So what does it tell you about light duration? Then the simplest way to say is there an effect when you vary the light duration? Is there an effect on protein synthesis? If so, what is it?
Yes, in presence of more light, the plants are going to make less of the protein.
You're saying of the protein. So why are you saying the? That indicates you're talking about one specific protein, but you just earlier said Ponceau was a non-specific protein.
Yeah, sorry, of all the proteins.
Okay, so in addition to experimental technique progress, whatever, this data indicates, and again, I'm going to leave it vague for class, but you should have something about the duration of light and its impact on total protein synthesis. Maybe you really look at it and say, well, it seems like, you know, when the plant doesn't have any light, there's more of this certain size bands, and when the plant has the regular light, the size of the proteins are very prominently 50 kD and smaller band size or smaller protein sizes. So whatever you and your group come up with, as long as it is supported by the data, you're good to go. Okay, great. So that covers that. So now let's go. Thank you, group 4. Okay, let's go to group 6. If you haven't yet described what the positive control is, like having used that phrase, the discussion is a brand new section. You definitely want to reiterate plants grown in a 12-hour photo period, parentheses, positive control, displayed, and so that you were just basically redefining what the positive control is. So positive control, let's go here. Let's start with what did you expect to see with the Ponceau staining for the positive control only? What specific thing did you expect to see for positive control?
So for the positive control, we were expecting to see a presence of bands, red bands in a column with thicker bands at the 55 kD mark. And I expected to see them also at the 13 kD mark, but I'm not sure if that's correct anymore since we learned that the 10 kD mark might not have fully separated.
But that shouldn't change your expectation, right? You should still expect to see it there. Now, whether you did or not, that may change, right? But that shouldn't change your expectation. So the specific data that group, sorry, I just forgot 6, talked about that is just specific for the positive control, because we kind of already talked about other things that showed up for all the samples. So I don't think we need to say that again, that we did expect to see a column or whatever you're saying, because we already talked about that expectation. And so we don't need to be redundant about it. Specifically for the positive control, we expected to see prominent, intense, obvious, notable, any of these words work, bands at approximately 55 and 13 kDs. Now I'm going to ask you why.
We would expect to see thicker bands at 55 kD and 13 kD because Rubisco is produced in the presence of light because it helps with photosynthesis.
How does 55 and 13 relate to Rubisco?
Oh, because those are the sizes of the large subunits and the small subunits.
Great, because these are the sizes of the subunits. And then I'd say, so. Then you'd say, well, Rubisco is a photosynthetic enzyme. And then I'd say, so. And you'd then say, and these plants were exposed to a 12-hour photo period, so 12 hours of light daily. So we expect them to perform photosynthesis. It's just kind of, this is your logic, and then you have some sort of reference at the end of that. Okay, perfect. This is exactly how you want to start it. Yeah. Okay, awesome. So what else do we need to talk about? So after doing the expectation and, the expectation and because, I'll stick with you really quickly. Did the data, where's my data? Did the data support that expectation? Yes or no?
Yes.
Okay. All right. So the data, yes. Here's where, and then we need to say, but here's where you want to bring up Why the band at about 10 kD? You say the data did in fact show that, whereas the positive control displayed very thick, noticeable bands at 50 and about 10 kD. However, the band at 10 kD may be ABC. Okay. That's 2 words. Okay. And this is kind of a writing team choice. You can make your conclusion about the positive control now, or you can explain the negative control and then make a conclusion about both of the controls in one sentence. So it's really up to you, and it's up to you and your team in terms of how it sounds. So here I'm going to pause on the conclusion, but I am going to say a conclusion at some point is needed. So I'll say conclusion here or after negative control. The choice is yours. Okay, really great job, room 6. So now we're going to go to our final room, and that's room 8. So room 8, let's go ahead and use the same kind of rationale here, or the same approach. Let's start with, what did you expect in terms of Ponceau? What did you expect to see that has not already been discussed?
Can you hear me?
I sure can.
Okay. So we expected to see a thinner band at 55 kD and 13 kD because without the presence of light, we wouldn't expect there to be photosynthesis, thus no rubisco.
Okay. Okay, which means no or little. You don't want to just say no. Here's why. If you just say no, then I'm going to say, then why did you expect to see thinner bands? Because that indicates rubisco's there, or it could be there. So you got to say no or low levels of rubisco. I would say very low, right? Because if you're expecting to see a thin band, that is equivalent to saying you expect to see proteins that are of the same size of rubisco, just in small amounts. But then you just said, if no light is shown, no rubisco. Okay, so those two things contradict. Okay, so what the hell was I writing? No. Oh, little. Okay. 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 But we're really pointing at rubisco, but we got to remember that the Ponceau stain doesn't specifically stain rubisco. It stains all of them. So these expectations are based on your hypothesis, but then your conclusion, this is when you have to write the word may. Okay, so what do you think if you're looking at the positive control and the negative control? So here it is right here. Positive control and the negative control. Let me get rid of all this junk. What does Ponceau say about presence of light versus absence of light with respect to maybe rubisco?
I would say, looking at the Ponceau stain and the negative and positive controls, that it doesn't necessarily support the hypothesis that light would affect necessarily just rubisco, but maybe all protein synthesis.
Okay, so we already made that conclusion up here, right? So that's all proteins, but you can make a possible, like it's not super well supported, but you can make a somewhat supported conclusion about rubisco. So if you look at this right here, what would you say? Oh, you know what? This Ponceau stain might tell me something about rubisco. What is it maybe telling you?
There is a slightly thicker band at the 50 kD.
So what does that tell you about rubisco in the positive plants versus the negative plants?
That there may be a higher presence of rubisco in the positive control than in the 60-minute photo period or the negative control.
We're just looking at the controls. We haven't even talked about this.
Okay,
Pretend
then-
this doesn't exist.
Then there's a lower percentage or lower concentration of rubisco in the negative control than in the positive.
Yes, and instead of using the word concentration, because we don't actually have a value for concentration, we say expression levels. Okay, because concentration would be 3 millimolars, 50 mg per mil. We don't have those numbers, so we only have more expression or less expression. But yes, that's what the Ponceau data possibly suggests. This data, or the data for control plants, possibly suggests, or likely, in this case it's probably a likely suggests that plants grown in a 12-hour photo period have more, let's say opposite, because we want to compare the negative control to the positive control, possibly suggests that plants grown in the absence of light have much less rubisco or have a lower rubisco expression compared to those grown in a 12-hour photo period. As expected, the data likely suggests that rubisco expression is lower in plants exposed to no light versus those exposed to 12-hour photo period. And that is establishing for your reader, hey, our controls worked the way that we expected them to. This is your conclusion for both of the controls. So we used this option here, and you have made a conclusion that satisfies both controls. You can absolutely do it the other way too, saying the data here suggests that plants grown in a 12-hour photo period have a very abundant, can possibly have a very abundant level of rubisco expression. Then you move on to the negative control, blah, 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 here's what we expect, da, 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 da. And similar to the positive control, our data supports our expectations such that plants grown in the absence of light likely make a very low expression of rubisco, okay? As evidenced by the thin band at 50 kD. So all you need here is 1 or 2 little specific details about the data. And then you say figure whatever B for the expected, figure whatever A. Every single time. Figure whatever A, figure whatever B. Don't just say, and yes, the data supported that. One specific tiny detail or one recap of a trend. That's all you need. Okay. All right, everyone, great job. Questions so far? This is a really good discussion. Also, I'm feeling 500 bajillion times better than I did yesterday. Okay, no questions. Cool. I want to leave this one for you as a team, because I think that we've spent a lot of time going through in detail how to explain these things here. And just recall that you can now look at how much stuff we have. Okay. How much stuff we have for similar data, positive, negative control. We haven't even started talking about the final plants right here. So this might be the place to remind you, it's definitely okay to break this down into paragraphs. Okay, there's a paragraph, and maybe we could do one more. There's a paragraph, And then in your final paragraph, you're focusing in on your testable sample. So I think I can get rid of this now or move it. Okay. All right, so we'll just say the 60-minute light. And if you are going to make them separate paragraphs, just remember you need a transition and then a topic sentence for the next paragraph. So I'm going to leave this up to you. Okay. All right. Last but not least, I'm going to go back to, I don't know. If I didn't call your group, can you just say yes in the chat? I think I called everybody. Okay. So last but not least, I'm going to call you on the spot. So let me see the groups. The one I was very, very impressed with their transition and topics. Room 3. So this I'm going to ask Greg to speak if you can. So Greg, if you can unmute yourself. If you can't, go ahead, Avery. But I would like for this last team to just wrap us up for today and tell us what the transition sentence was to end this paragraph. So we are ending the paragraph about Ponceau discussion, and we are going to head into immunoblotting. And then the next sentence should be the topic sentence for the immunoblot. So group 3, whoever can unmute themselves, it'd be better if it's the other person, but if you can't, that's okay too.
All right, yeah, no worries. Let me just go back to my page. So our transition was to really understand what the presence of rubisco is in each sample. We have to separate those proteins from the other non-specific proteins using immunoblotting.
Okay, so pause there. So good draft, but a couple things that I think we definitely need to fix. And I think here is where we might want to reestablish why we think rubisco might be changing. So I think it might be nice to say, because we just finished talking about 60 minutes of light. So to truly understand how a brief 60-minute of light exposure affects rubisco specifically, right? Because before Ponceau, we're just kind of like gathering clues, but we can't really say. Right? Then you say an immunoblot was performed, conducted, whatever. I think that's great. Okay, so we want to make sure that we know we're not separating out rubisco. So when people say separate out in science, that literally indicates somehow you are getting rid of the rest of the proteins from the surface of that nitrocellulose membrane. And the only thing remaining on that membrane is rubisco proteins, whether it's a large subunit or the small subunit. And that's not literally what's happening. So you can't say that. So science writing is quite literal. So you don't want to say things like isolate. If you want to specifically look at a certain protein, you can do that because that's essentially what you are doing. So just be careful that you're not isolating or separating out because you're not doing that. All the proteins are still there. Okay, so maybe your sentence needs a little bit of modifications. Okay.
Okay.
Let's see. You can't speak of your connect. Okay, don't worry. Yeah, specifically detect. That's a great phrase, Natalie. Awesome. So let's go with your topic sentence. So how do you start this last section of your discussion? That's pretty much like the fireworks of this entire study. So what is the topic sentence that you have next?
I wrote immunoblotting is a technique used to block non-specific proteins and the membrane blot from being detected in the final stain to effectively allow us to see rubisco. But since talking about like what we talked about in the results, I definitely want to revise it.
Okay, Yeah, and then I will point out that immunoblotting itself doesn't block the membrane, it's the blocking solution. And that's something that you don't really have to go into a lot of detail about, because I think most scientists know that blocking solution blocks the membrane from non-specific binding of the antibody. So the topic sentence, let's see. Okay, so thank you to this group. Who else had a really good? I can't remember, so let's just try. How about let's go back to room 8. Room 8, can you tell us what, oh no, we just heard from room 8. How about, oh, let's see our group of 3. How about room 1? So room 1, somebody different, go ahead and unmute yourself and tell me what the topic sentence for your immunoblot discussion paragraphs would be.
Me and Avery were in the same group when we did the thing, so I actually have the same exact sentence written, but I don't know, maybe Heidi, do you have any other ideas to add to it?
Yeah, 1 second. Let me pull up my other. Okay. So we have.
And it can be pretty similar to this right here. So you can use that as a jumping off point.
Okay.
Maybe just a little bit more information, right? But you can even use that, switch some words around. So what are just some of the phrases that maybe we can consider when doing this last topic sentence?
Some phrases. We use it to isolate and identify the expression of rubisco in the P. sativum samples. That's what we have.
Okay, so we're not isolating, but you are, you can, like, this is your last couple. This is like almost like your final impression. So if you want to bring up, and I think that might be a nice idea, if you want to bring up one more time and be like super formal, and instead of calling them peas or plants or any of these things, you can bring up, sure, the actual species name. You can bring up, I think once again, even though you did it in the results, I think it's important to remind people that you're specifically going to be looking at the large subunit. And why is that important? Because now you're focusing in just on that band at about 50 kD. And maybe that is something else that you can bring into your topic sentence, so that there's no question on the immunoblot which of the bands that you should be looking at. Here, unlike doing it in the results section, here is where maybe you want to talk about the detection being a colorimetric, an enzymatic colorimetric color reaction. So it's a color reaction, something about an enzyme, and you can keep it pretty simple. So you're going to be looking at a purple, right? A purple color precipitate. You can use any of those phrases to really kind of start the next, I don't know, 1 to 3 paragraphs. Okay. And I think that that covers it, everyone. And you can obviously see that even though, let me try this way. Okay. So even though in the results, the paragraph for Ponceau was only 5 to 6 sentences, 1 paragraph, 5 to 6 sentences. For the discussion, here's your copy paste. Here's the start of the paragraph, topic sentence, paragraph number 1, transition, topic, paragraph, maybe even 2 or 3, but probably just paragraph 2. Transition, topic, paragraph number 3, final transition, And then here's where you start talking about immunoglobulin. So one paragraph over here, 1, 2, 3, possibly 4 paragraphs for the discussion. That is the balance between results and discussion. I'm not saying the paragraphs need to be a page long. That's a bad paragraph, but concise, organized, consistent paragraphs. Boom, you have communicated your science. If someone doesn't get it after this, they're not going to get it. Okay, is there anyone that has any questions about this? Okay, great job, everyone. Thank you for being here. I don't remember off the top of my head who's normally in Wednesday and who's in Thursday, but if you are in the Thursday lab and you came today, welcome, and I hope this was helpful. If there's no one in the Thursday lab, well then, good job to everyone in the Wednesday lab who came. And I hope that you are feeling good about the report. I hope that you are feeling good about the rest of your day and feel like you have the time to study. There are some questions. Megan.
I had a couple questions. First question, this may be a redundant thing, and I'm assuming it is, but do we need to include in our discussion the how we talked about the data from the experimental control and the negative control and all that? Do we need to include at all the molecular weight markers? Any information on them?
I think that's a good thing you want to reference. So I think that's more of a results thing. So less of a discussion, more of a results thing, where if you're talking about the first time you talk about these ranges, you can simply throw in a phrase that says, as compared to the molecular weight marker in lane 1. That's it. Yeah. And then when you show this, if you do crop this out, right, you'll have your computer generated text boxes, but maybe you have them labeled 1, 2, 3, 4, or maybe you have them M plus minus 60, however you want to do it. It doesn't matter. But in the caption, you want to present the molecular weight marker by the official product name, like the same way you did it in the method section. You want to present this in the caption for the Ponceau, and again in the caption for the immunoblot. Even if you're like, well, I already did it, they'll know. Not necessarily, because what if they only look at the immunoblot and be like, what marker did they use? Oh my God, these people are making me go back to the method section to find this information. Couldn't they just put it in the caption? So your goal as a writer is to make it as easy for the reader as possible. Okay, any more questions?
I have one more to add to mine, sorry. So just to confirm, in regards to the topic sentence for the discussion, the last part we talked about the immunoblotting, would you be super specific about like the primary antibody there and the blocking solution?
Nah, I think just forget the blocking solution for the topic, because remember what a topic sentence is, all of the other sentences have to support that, and the blocking is just one minor step. Like when you're talking about purple color, that's not because of the blocking. Right, so your topic sentence, I think leave the blocking out. Like if you wanted to maybe have the second sentence say, before immunoblotting with antibodies, the membrane was blocked with 5% milk to, was incubated with 5% block to 5% milk solution. What is it? Milk in TBST to block non-specific binding of the antibodies. You can do something like that, but keep it super brief, because the discussion should not focus on methods. So if you can be concise and write about maybe like the list of the things that you did in one sentence, then go ahead and add it.
Okay, thank you.
Sure. Arlene.
That was basically my question, as I specifically detecting like Rubisco, whether we should include like the substrate and how that binds to alkaline phosphatase in the secondary antibody. That's kind of what.
Yeah, just keep it super concise. So, you have your topic sentence. The membrane was first incubated with 5% milk and TBST to block the membrane. Then a series of antibodies were used. You've already talked about the primary. So, or you can even say like after primary antibody incubation. A secondary antibody linked to alkaline phosphatase was used in order to colorimetrically detect Rubisco expression.
Okay.
That's it. And that's all that you really need in discussion, because this is not a methods paper, it's a research paper. Now, if you were testing a new antibody, then yeah, you go into all of that explanation. Okay, now I just want to remind everyone, we just did this for the Ponceau. So you have to do the same thing. So think of me, all these questions that I'm asking you and the order that I'm putting stuff in. If you haven't already, make sure if you need to revise your own outline before you turn it into a final draft, so you have this stuff, do it so that it's nice and organized. So I hope that this workshop allows, with the previous workshops, not only from the Western Blot, but also from TLC, really gives you a full representation of all that goes into writing a proper formal lab report. And so to keep this in mind, the next lab activity, module, whatever you want to call it, it's not a lab report, it's something called a grant proposal. So you'll see some similarities, but you'll definitely see some new stuff. So this is kind of your last lab report. Yay, I'm glad that they're helpful. I will tell if they're helpful by when I read the reports. That's how I'll tell if they're helpful or not. So I wish you all well, not luck. I wish you well studying today, and make sure your notes are nice and organized. Your half page of note is nice and organized for tomorrow. Do not forget to submit it to Canvas. I'm not going to chase you down this time. So if you didn't turn it in, but I can see that you used it, you're gonna get a point deduction. You guys have to be responsible for these notes. You're the first class that has ever had the opportunity for notes. Don't ruin it for the future. Okay, so have a great day, everyone, and I will see you all the lecture stuff. I'm getting the last lecture, lecture 14. I'm converting it to a YouTube URL now. After the test, you'll be ready for a break, but then you'll have all the information you need to get ready for next week. Have a great day. See you soon. Bye-bye.